And it's Cocktails and Fishtales time, everybody. I'm so excited already because we have such wonderful presenters and really good friends, Ashley and Fran, here tonight. So I hope you're excited for a fun presentation to learn more about larval crab monitoring here in the Puget Sound. But before we get to that, we of course like to give everybody a little time to settle in, log on. And while we're doing that, we ask our presenters to share with us a cocktail and fishtail that they enjoy. So I'll let you two rock, paper, scissors to determine who wants to share first. But
you want to go? I'll go first.
Great, Ashley's on.
Hi everyone, my name's Ashley, and my cocktail of choice would be a tequila sunrise. And then my fishtail, so when I was getting dive certified actually with Fran here, we found an octopus just crawling on the floor, and he crawled into our hand and then he inked away on me. So that's probably one of my favorite stories because octopuses are one of my favorite animals.
That is
Hi everyone, I am Fran, and let's see, favorite cocktail. Let's go with a mojito. Be cool if we could make like a seaweed mojito.
Like you both are picking such like delightfully summery drinks. I'm like, a week ago it was August in October, so
yeah, we're still there mentally maybe. And a fishtail, man, I have so many fishtails. I feel like, I guess one of the ones that resonates is when I was getting my navigation certification scuba diving. It was a night dive. And I was out there and I was having to go a certain direction, and you can't not go that direction or else you don't pass your class. And I had never seen a harbor seal. And of course, as I turned my 90 degrees, I'm underwater in the dark. There is a harbor seal just sitting right there. I'm face to face with him. I was like, oh, hello. And so it was quite a spook for me, but they're like super friendly, like dogs.
What a lovely first harbor seal encounter.
It was quite spooky.
Well, for those of you tuning in, you're welcome to share what fun beverage you're partaking in this evening. And if you want to throw in your favorite fishtail, we'll take stories as well. But the comment section is yours to use for asking questions, saying woohoo, doing, interacting with us tonight as we do our cocktails and fishtails presentation. So use that comment section. We'll make sure that all your questions get answered at the end of Fran and Ashley's presentation. So with that, I guess I'm just so excited that you both are here. I would like to point out that these two have been a critical part of the Harbor WildWatch beach monitoring team, especially during the pandemic. So they came out with us in the dark of the night, and I know Ashley, and I think Fran too, survived the mud of Austin Estuary. And so there's been a lot of good science done between these two, and they're of course magical scientists in their own rights and doing awesome things like the crab trap monitoring surveys. So we're excited to have them here to help us learn more about what it is when we hear these things called crab traps out in the Puget Sound, and what is this larval monitoring all about? So I'll turn it over to the two of you to tell us some more.
Thank you, Stina. And just a forewarning, we do have two dogs here, so if you hear some barking, they just wanted to contribute also. But Fran, we'll take it away.
Awesome. Can you see everything over there, Stina?
Looking good, you too?
Is that fine? I'm not sure. Okay, we can go from here. Awesome. So good evening, everyone. My name is Francesca Marvin.
And I'm Ashley Benson.
And tonight we're going to talk to you all about larval Dungeness crab study that we've been part of for the last couple of years. So who are we? Both Ashley and I are lab techs for the University of Washington Tacoma Becker Lab. The Becker Lab's principal investigator is Bonnie Becker, who is the founder and leader of the Becker Lab since 2006. The Becker Lab is a marine invertebrates lab that specializes in shellfish. We are comprised of mostly UWT students and alumni. Our lab provides students an opportunity to get valuable experience in the lab and field conducting research. Most of them will base their capstone research off what they do in our lab.
So as Fran mentioned, the lab has been around since 2006. And so over those years, we've had many different research projects that we've focused on. This is just images of a few of the ones throughout the past that we've worked on. So some of them being the geoduck settlement and larval distribution, Olympia oyster restoration work, marine sediment monitoring, and then the most recent one being the larval crab study. So with that larval crab study, we partner with the Pacific Northwest Crab Research Group, PCRG for short. This research group was founded in 2018, compiled of a collaborative group of tribal, state, and government partners looking to address critical research gaps within the Dungeness crab population. So this is important because Dungeness crab is one of the most highly utilized marine species on the West Coast of North America, playing a relevant cultural, ecological, and economic role throughout the state. So the Washington State Dungeness crab fishery has become progressively more active over the past 30 years, with around 10 million pounds, with around 10 million pounds of crab harvested annually. While the biology and ecology of Dungeness crab is relatively well understood compared to other marine invertebrates, many questions regarding how Dungeness crab populations vary over time and across space, and what environmental variables influence different life stages is still in question. And then just to note, the main mission of PCRG is to promote and support sustainable Dungeness crab populations in the Pacific Northwest. So PCRG has a few different research projects that they work on. The one that we're going to talk about today is their larval crab study. The research question that we're looking to answer is, what is the best predictor of adult crab biomass? So this question was determined by PCRG members to be our top research priority. And this question came about based on a paper written by Dr. Alan Shanks from the University of Oregon, who provided evidence that larval crab abundance can predict adult crab abundance 4 years after the larvae are caught. So based off of this knowledge, PCRG built this model, decided to build off this model and determine whether it could work in Washington State by developing a statewide larval crab study using light traps deployed on docks and piers to capture Dungeness crab larvae. And if you want to learn more about PCRG's other programs, I've linked it at the bottom there. It's pnwcrab.com/programs.
Awesome. So I'm going to talk to you guys a little bit about the Dungeness crab life cycle. So in the larval light trap, we catch the first 3 life stages of Dungeness crab: zoea, megalopy, and juvenile instar. Both the first and second stage, zoea and megalopy, live pelagically. This means they're suspended in the water column, whereas the third life stage, juvenile instars, settle out on the ocean floor. The reason we see juvenile instars in the trap is because they've actually molted from their megalopy stage into being a juvenile instar while in our trap. When a crab is in its zoeal phase, it's extremely hard to identify what species it is, but megalopy larval stage is much easier to identify, which is why they are the life stage that we end up focusing on. Here's just a fun video to show you guys what Dungeness, zoea, and megalopy look like under the microscope. Kind of creepy looking, kind of cute, but. So you might be wondering, what is a light trap? On the left is an image of the light trap put together and ready to be deployed. A light trap is basically a water bottle with funnels on the side, a buoyant lid, and a light in the center that turns on at sunset and off at sunrise. At the bottom of the trap is a cod end that functions as a strainer, allowing the water to drain while capturing any organisms that entered the trap. Finally, there's a weight attached to the bottom of the trap to keep it perpendicular in the water. The way that this light trap works is at sunset, the light turns on. Larval crab and other larvae exhibit positive phototaxis. This means that they are attracted to light. We take advantage of this behavior with these traps, which are especially effective at capturing megalopy. At sunrise, the light turns off, We pull the trap out of the water, capturing the organisms that entered throughout the night. The trap is checked it's checked 4 days a week, typically Monday, Wednesday, Thursday, and Saturday. The fishing season normally runs from approximately April to September, but it's dictated by when we're seeing these Dungeness crab larvae. This is what a common catch looks like for us. We put the sample in a white tub with an aerator to keep the water from going hypoxic so that the organisms stay alive. Then we sort and count our sample. And then here's a little video for you guys to bring you virtually to our trap. Ooh. Ah. Awesome. Move from that. So then we sort the different species into beakers, identifying, counting, and recording all the organisms. We measure and record the carapace width, height, the total height, and the total height of the Dungeness megalope. PCRG uses this data to help assist with the question of if there's more than one cohort and track changes in larval sizes throughout the year. All right, bycatch. We get a wide range of bycatch in our traps. The first organism that's probably catching your eye is some absolutely adorable larval octopuses in the far left video. We also get larval Pacific spiny lumpsuckers, clingfish, salmon. In the hand, you're seeing a ctenophore jellyfish. We also see amphipods, ostracods, polychaetes, of course, larval Dungeness crabs, other species of crabs, and countless other zooplankton. This definitely makes for no boring days at our traps. Dungeness crab is one of the most highly utilized marine species in the Pacific Northwest, fulfilling cultural, ecological, and economic roles throughout the region. Despite the clear importance of this species, Dungeness crab populations face many potential threats, ranging from global issues such as ocean acidification, warming sea surface temperatures, to more localized issues like coastal development and also fishing impacts. As these threats intensify, scientists and resource managers will need to develop and implement adaptive responses to sustain the species and the biological systems that they depend on. Working off Dr. Shank's research, PCRG will be able to supply the fisheries with accurate adult population abundance. They are also filling in key unknown information about Dungeness crab in their larval stages, which may assist in explaining the disappearance of Dungeness crab in the southern Puget Sound.
So this map here shows the location of all of our crab traps across the Salish Sea. So there are approximately 20 traps in the US Southern Salish Sea, and we have 25 in the Canadian Northern Salish Sea. The two traps that we participate in are indicated there by that red arrow. So we have one at Redondo Beach with our partners at the Highline Marine Science and Technology Center, and then the other in Tacoma at Point Defiance, working with Sammy in the Point Defiance Zoo and Aquarium. Here are all the many partners that we work with, and there's many more that weren't listed here. Some of them being like National Fishery and Conservation Center, Northwest Indian Fishery and Commission, the Nisqually Research and Nature Center, amongst many others. And if you're interested in becoming a volunteer or wanting to learn more about the crab research group, you can reach out to myself or Fran for any additional information or how to become a partner.
And I'd also definitely recommend looking into the Dr. Alan Shank's work if this interests you at all. He's kind of the spearhead of all of this, an amazing researcher, and PCRG is really basing all of their methods off of what he's already done in Oregon. So super interesting. He's got lots of good papers out there. And that's all from us, Dina. Back to you to open it up for any questions.
Awesome. Well, thank you both so much. We have folks tuning in from Los Angeles and Tacoma and Gig Harbor. Lots of love coming in through the chat for you both. But are there any questions? Now is definitely the time to put those questions in the chat so we can have Ashley and Fran answer those live. My first question, when I saw that harbor seal, I'm like, is that the same harbor seal, Fran?
This is not, probably not. This one's down at actually one of our traps, the Point Defiance trap. I had that, I wasn't there that day, but they sent in a picture in our little group chat. And where I saw mine was actually up at Alki. There are always some at Alki, I learned.
Yeah. I think the nice thing about doing field work is even though you're like looking for something specific, there's always so much to see. So you never know what you might cross paths with.
I know. Are you a spiny lump sucker lover like
My
me?
gosh, yes.
When I saw the larval one, I was just like, it was done. That was it.
I think my heart exploded. It's the cutest thing I've ever seen.
Won't get better than that.
All right, so I'm gonna peek over here and see what we have going on. It looks like we have Jake asking, how was the trap created? I'll follow up with like, yeah, is this something you have to put together, or is oh man,
Yes,
Puget Sound crab, or what's the crab group letter combination again?
so there are so many different traps. As Ashley had mentioned, there's about like 50 traps. And then each trap location is responsible for making and creating their trap, but we all follow the same protocol, which has some wiggle room in it, because some of the traps are put off a dock that you can just walk down to. Some of them you have to like grappling hook it up. So some of these need certain special modifications, but the traps are made by us. Every season we'll remake the traps over and over and over again. As you could guess, things tend to break down quite fast in salt water or get very algae covered and barnacle covered. And so hopefully, Jake, that answers your question.
Yeah, it seems like I liked the clever use of the like Culligan water container bottle. It seems, how cost effective is it to make a trap? I'm guessing the most expensive part is the light, but is there a special light you use too? Maybe we'll follow up there.
The lights and the batteries are pretty spendy. I'd say like the budget for the trap, at least when we have the sheet of like materials needed, it's around $400. But that was before price increases and everything, so it can be a little more, but that's also including batteries and everything, which are probably the most expensive part.
And you were able to find those water jugs for free, right?
I was,
You go and you ask very nicely. Certain places will give you them for free.
Love that. Let's see. It looks like so Jake is furthermore curious as to what is the process of making those traps.
So the process is, it takes a lot longer than you would think. It takes longer than we think every year. But you start with your jug and you have to drill a circular bit into the side of the jug. And this requires you strapping the jug down to a chair or something where it won't move. And
thanks for choosing a chair and not like Ashley.
Yeah, well, it's definitely gone between the legs before, and then the bit will slip, and then you're like, never again will I do that. And so you drill the holes in it, and then you'll end up just attaching the funnels with like either a silicone, which will end up coming off, or zip ties. Thank you. The Pelican box is a little bit more of a pain with all the electrical in it, because you just drill the holes and then you glue on certain fittings and toilet flanges and such. And then classic scientists just using all sorts of random materials. But then the actual lighting unit takes quite a bit to be able to get that to work. They didn't really give a straight up method for that. And so we kind of had to play around with it to figure out how we could get the light to actually work and the timer to be able to connect the light and the battery to that. So the first time we got it to light up was very, very joyous moment. Yes, me and Hidoji actually were out on the dock for like 5 hours.
Well, good job using your tools. I'm impressed at the amount of plumbing materials that often gets co-opted into marine biology research.
Yes.
So cheers to the flange.
Cheers to the flange, man.
We have Wendy tuning in wondering, who do you work with in the Salish Sea in Canada?
It's a great question. I'm not entirely familiar with the exact organization that we partner with. We only recently started partnering with them. I believe it was this year was the first year we had partners in Canada. But yeah, I'm not sure off the top of my head what organization they're exactly from.
I can drop that in the chat, and I see that Lindsay's in the chat too, and so she definitely does know their name. I forgot it right now, it's slipping my mind, but we were talking about it this morning. Oh, there we go. The Hawkeye Institute is who we partner with, and they're in charge of all of the other traps that are in the northern Salish Sea, so the Canadian traps.
Fantastic. I have a question about, I just got to meet some of the team from Puget Sound Restoration Fund, and they were talking about crab traps, and I was like, oh, do you know Fran and Ashley? And they're like, yeah. So what is the connection between Puget Sound Restoration Fund and PCRG?
I believe, I don't know exactly, but to the best of my knowledge, I know that one of our lab members also works there. And so if you know Mackenzie,
Got to meet Emily, but
I think that she's either interning or working there. I'm pretty sure she's working there. But that is the only link that I know.
Sweet.
And other than that, I think that they do have a trap too, but I think it's just run by her.
Awesome. Cool. Ash is wondering, what do you hope the future looks like for this type of work? And I'll maybe tie that in. My mom's tuning in and she wants to know, how is the crab population and what are their biggest threats? So kind of that, how's it looking and what does the future hold for these fine little tiny crab friends?
Yeah, I think I can answer the first part of what we hope that. So right now there's a data gap within this Dungeness crab stage. Yeah, so we don't know what's happening to them from when they are larvae to when they're able to be harvested 4 years later. So we're looking to bridge that gap, see what's happening to the population, and just gain some baseline data on the larval crab population, since there is none at the moment.
And to add on that, a lot of the unknown is going to answer a lot of the unknown. I hate to say that. Let me restate this. Knowing more about the Dungeness crab larva is going to allow us to answer a lot more questions on what's going on with our population, because right now we have the fisheries closed in the southern Puget Sound. We don't know why. We don't know if it's disease. We don't necessarily think it was just disease, if it's just no habitat left. If it is overfishing, there's like many different variables that it could be, but we don't know until we do the research. And so right now we are seeing Dungeness crab larvae down in the southern Salish Sea. So why aren't they settling out? Why aren't they, residing there as adults? Or are they dying before they become adults? And so that is a lot of the questions that we seek to answer. And PCRG has a lot of side studies aside from just presence, absence, and then also the different sizes, the measurements that we take. They're also doing genetic work, and then they're also looking at temperature loggers in all the areas that our traps are at. And so being able to get more data, we're still such a new organization, or they are. And so being able to compare that over the years is definitely going to be able to allow us to answer that question and maybe create more habitat for Dungeness crab in the southern Salish Sea if that is the problem.
Fantastic. Yeah, I think I guess that you're mentioning the larval, getting a look at the larva, and then that can give you a 4-year prediction. It's kind of interesting. So I guess it's good to see, are you seeing lots of larval Dungeness crab in your light traps currently?
are seeing larval Dungeness crab. It's hard to say what is a lot when you don't have a baseline.
Sure.
And so creating the baseline, where we see much less down in the southern Salish Sea compared to like right at the inlet or in the northern Salish Sea. But yeah, and I don't know if it was already stated, but along with like your mom wondering more about the adult population, this study is being done so that we can have a method in place to be able to predict the adult population 4 years out. And so then we will not have any problems in terms of like overfishing. And right now we don't really use a good model, and we also don't have a good baseline of knowing how many adults we have. And so then it's just based off of size. We could be overfishing them and depleting it further. And this Alan Shanks used this model in Oregon, and it worked very well. All the fishers ended up coming to him before it was implemented, asking him like, how many should we take? Where should we take from? All of that. And so it's exciting because Washington will soon have a very, well supported method to be able to get our population abundance numbers and our fishing numbers.
Awesome. We have a Jacob Marvin tuning in to ask, how long have you been doing this?
So this is our second season.
Okay.
But PCRG has been doing it at different traps for the last 6 or 7 years.
They were founded in 2018, so it's only been 4 years.
Well, and so then to add to that question, what trends do you find within that kind of short time frame? Have you seen any trends? Like, are there times of year that there seem to be more larval crab, or
peak season? Is it July?
It's around July.
It's around July.
So right after our heat wave last year, kind of during it even, we had this huge spike of megalopy, and we saw much more megalopy last year than we did this year. But it went from seeing, maybe like a handful, so 10 to 20 of them in the trap when we checked it, to just hundreds overflowing, like coming down onto the dock, trying to pick them up to save every megalopy. So we do notice a peak, and that is a standard throughout all of the other traps too. We're all seeing this peak all around the same time each year, and it is kind of around July. And we also saw a peak this year, but it was just a lot less. And so we didn't breach 100 at either of our traps, but we have yet to hear the data from the other traps. So I'm sure we're normally one of the lower numbers in terms of how many we see. So hopefully other people saw more.
Which, when it comes to counting tiny things, you're kind of like, phew. We have Rachel Easton tuning in with some great questions, wondering how ocean acidification might impact the larval crabs, as well as maybe the adults. And then wondering if you can notice European green crab larvae. Larval European green crab. Is that something you're on the lookout
I
for?
wish we could see, and we have. Well, I'm happy we haven't seen them. I don't want to make that statement, but they are very, 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 very small compared to Dungeness, and we are seeing other larvae, but they're so small that we necessarily wouldn't know we're catching them. Dungeness crab megalopy are the largest of all the megalopy, so they're like almost a pinky nail. And whereas other megalopy can be, as small as like a pen point. And so there definitely can be studies branched off this just to see if there's DNA of these green crab, environmental DNA, in the water or in the area. And that's something that people are thinking about. But I do know that with the European green crab, their settlement, the smell of it will deter the settlement of Dungeness crab megalopy. So that is something to think about in terms of, maybe there being a lack of habitat for these Dungeness crab. But we also aren't necessarily seeing European green crab in the southern Salish Sea. So hopefully it stays that way.
Yeah, totally. That's definitely something Harbor WildWatch is keeping an eye out for. Not to be confused with our beloved green shore crab and other, sometimes the purple shore crab that has its green morph, right? We like those guys. So before you go crushing crabs, make sure you know what you're doing. That's my public service announcement for you all. I think we have Lindsay tuning in, and I'm like trying to I keep losing track of my question that I was about to ask you. But yes, so Lindsay Overstreet working with y'all and saying that Puget Sound Restoration Fund is the main coordinator of PCRG, but I'm not going to quote Lindsay on that.
Oh, yes.
That's, even though I kind of just did. Sorry, Lindsay. Thanks for sharing that. And then, oh, where did it go? The scroll. Oh yes, Lindsay in the trend question is adding to that this year there was kind of a late June, early July at PD, which I'm guessing is Point Defiance.
Yep, Point Defiance shop.
Not sure about the Mast Center.
Mast Center, it was all about the same. I was from one day a week at each of the traps. It's fun to see this year. We'll see if we add another trap or not next year, but two was fun to be able to compare what I saw day to day.
Oh, totally. And is that, yeah, so on a weekly check.
And so it'll be 4 days a week, but yeah, I just went to each trap normally one time each week.
Gotcha. And is the sampling year round? Sorry if I missed that.
So the Mast Center is now doing sampling year round. This is like aside from PCRG, just to see what are the different trends of, larvae throughout the year. And that's exciting. And I can't wait to see what they see. And I heard that like right after the larval crabbing season, that there was just tons of shrimp, just like shrimp season apparently is right after crab season. At least on the larval level. And so that's not normally something that PCRG does is year round, but Mast Center is open to having volunteers for that. And then this is the first year it's being spearheaded. So we'll see what comes of that.
Yeah, it seems like, I don't know, just like such a beautiful hands-on education tool to get people stoked about the planktonic life that is abundant in our waters. And again, yeah, seeing all the cool things like baby spiny lumpsuckers, and that fits in nicely with Jacob's question. Did you catch any fun one-off stuff in the traps?
Go back to great question. Yeah, let's pull up some of the fun one-off stuff we caught in the traps. I mean, the octopus was pretty cool. I was not there that day, and I was trying to make it down once I heard it in our messaging. I tried to make it down to the trap in time, but they had to put them back in the water so they wouldn't die. So as you can see, is our screen?
Your screen is sharing, yeah.
Awesome. On the far left, you can see the octopuses, and then the Pacific spiny lumpsucker. These were some of my favorites, which is why I decided to add them to the slide. And the clingfish. I mean, these are just absolutely itty bitty. And then the ctenophore jellies are just always so fun. They refract light in a way where it looks like rainbows are running up and down their body when they're in the water. When they're out of the water, they kind of look like a grape without any skin. And then we're catching salmon, which actually eat megalope. We're catching a lot of like forage fish. We're catching the classic zooplankton and what were you gonna say?
Oh,
polychaete balls.
Polychaetes and shrimp and other things too. Yeah.
And the polychaete balls, man, those are just quite a sight to see. If you guys don't know what they are, it's just like this breeding ball where they like breed and they die and they're like breeding, and then it's just like this massive, ball of life and death, and it has quite a smell to it.
Fantastic. I wonder, that's like the sea slugs that smell like Jolly Rancher watermelon. I wonder if there's some sort of
smell like that.
Corey is tuning in, mentioning a little bit late. Oh, they're just telling us where they're logging in from. Thanks for watching from Hood to Now, Corey. I will say, for those of you who are just tuning in, the whole video will be posted at the end. So you can always go back and catch up with what you missed, and we'll post this on YouTube as well. So with that, I'm doing kind of the catch through, seeing if I missed any questions. It looks like we have one from Richard, wondering what is the habitat required and could temperature play a role? guessing for maybe more of the adult Dungeness crabs.
So in terms of adult, we don't know as much, just because our focus has been on larvae. But the habitat, we've been really wanting to and looking into mapping out and trying to find some funding to be able to have a researcher map that out. Normally kelp beds for adults, but in terms of the larvae and then where they settle for those first 2 years of life as juvenile instars, a lot of that is unknown. And so there hasn't been a lot of like mapping out where they are in their larval states until they're to that like mature age. And so that's definitely something we're interested in looking into, but no one's done it yet.
Fantastic. The wonders and questions.
Love them.
Lindsay is chiming in with the one-off edition of a weird looking baby flounder.
Yes.
So, yeah, I think when things are tiny, it's just extra fun and cute. All right. Well, I hope I caught everybody's.
I see one in here from Terrence Lee.
Oh, Terrence, fantastic, from the Nisqually Reach Nature Center.
Oh, lovely. Oh, and there it's back. He's asking if we saw multiple pulses of Dungies in our catch. And normally we just see one pulse. This year we're still processing the data, so it is yet to be known. But from my memory of like what we saw at the traps, it was just really one major pulse. And this year it wasn't that major in terms of like a rise in our numbers, but yeah, just one this year.
Which then maybe we'll ask Terrence, because I believe they do a larval crab trap as well. Did you see any pulses while you're doing your
Terrence? Let us know.
Flip the question around here. Lots of, I think maybe just another cool thing that I'm just noticing about this is just how many groups there are to make this work possible. I think
so many.
There you are. Your partnerships are bursting with folks. So
yeah, it's definitely a great thing that really a lot of different organizations can allow citizen scientists to jump on if they have like a lead, someone who can guide everybody, because it's not really hard work. It's just more knowing what to identify, how to identify, and then just recording that. And so that is a really cool part of this work, is it is very open to all different age levels, knowledge levels, all of it. And yeah.
Fantastic. I will say Terrence is saying, yes, they did see a pulse, which is why they asked the question.
Just one pulse or two?
How many pulses, Terrence?
How many? All of our data is due by the 31st, so I'm sure we'll all know soon.
Well, this sounds like this could be a follow-up Cocktails and Fishtales presentation in the future. Lots to be learned from these fantastic folks doing this amazing and what sounds like some pretty fun observations out in our larval communities of the Salish Sea. So I think, is there anything else the two of you would like to leave us with before we wrap up here? I know I have a couple Harbor WildWatch announcements to end on, but I'll let y'all have the last presentation word here.
I think that's about it. Yeah, reading Alan Shank's papers, that's my only piece of advice. If this work interested you at all, or if you want to open up a light trap, if you think you have the capacity for it and you're at an organization or whatever it is, of course, email me or Ashley. We'd love to help get you connected to the right people to start one up.
I feel like Harbor WildWatch capacity is low right now. I was like, I've been emailing new volunteers being like, welcome, it's our slow season and da-da-da-da. But I'm like, I'm so sorry, I have lied. It's not slow at all. There is so much going on. So if we have any volunteers tuning in, there's a lot of programs, but yeah, what does it take to have a larval crab trap?
Just takes the
Definitely
willpower and
consistent
volunteers.
too, yeah, because it does need to be checked 4 times a week at a certain time. So making sure you have someone who's able to consistently be there to check it.
Okay, so 4 times a week at a certain time, probably access to a dock.
Some type of dock, I was gonna say, or pier to be able to attach it to.
Supplies. And then are you looking at things with a dissecting scope or just eyeballs?
Both.
Cool.
It is really more like the only thing required by PCRG is tracking the Dungeness, which you can see with your naked eye very easily. And then everything else is just, whether it's student research or personal interest or the organization's interest, it's all optional. So you could make it as much or less as you'd want it to be.
That's the other joy of community science is like my cat being like, you're doing something important. Yeah, just kind of being able to tag on those extra little notices. So yeah, I'm like harmful algal blooms, and they're probably not attracted to light, but there's a lot to do with tiny water things, I'm sure. Just those notices. So again, round of applause for you all. For those of you tuning in, if you want to write in the comments all your kind, loving words of joy and celebration for a fantastic presentation for these two. So with that, lots of fun stuff going on. Hope you can get involved. And again, if you're interested in more information on this crab research, happy to connect you with these two, and they can connect you with more people as well. So teamwork makes the dream work with these fine folks. So thank you again, Ashley and Fran, so much for joining us for the lovely October Cocktails and Fishtales tonight.
Thank you for having us, Dina.
Yay! With that, again, just lots of love coming in through the comments. So make sure you take a peek at those at the end of this, and we'll go ahead and sign off here. So as always, learn, have fun, everybody, and have a good night.
Bye.
Bye.
Thanks, Gina.